Assalamu alaikum, dear students. Thank you so much for following the sequence of cell cycle or cell division. Let's start today's video, cell cycle part 3, meiosis. And let's start with a question. Have you wondered why children do not look exactly like their parents? Or why some siblings look so different from each other? The answer lies within meiosis cell division, which generates the gametes for reproduction. Meiosis is a process by which haploid cells are produced from a diploid cell. For this to occur, chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells. Pay attention, not identical cells, but in order to produce genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonad of male and females. For a successful reduction in the number of chromosomes to occur in the newly haploid daughter cells, 2 rounds of divisions are necessary to occur. These divisions are termed as meiosis 1 and meiosis 2. Although process of mitosis and meiosis look similar, but there are few major differences. One obvious difference is that mitosis result, as we studied that we result in mitosis by 2 identical cells, but in meiosis we will result in 4 haploid cells, which are genetically called unique cells. So in order to understand the meiosis in more details, let's talk about it. First of all, let's talk about significance of meiosis. If you remember, we studied about the significance of mitosis, that it was used in order to produce 2 identical cells, in order to use for growth and also for replacing worn out cells. And the final one that were the major purpose of mitosis was its usage in asexual reproduction. But in meiosis, we will study that sexual reproduction leads to genetic variation among the offsprings. That is the main cause of meiosis. It means meiosis makes genetic variation possible in 3 ways. First, crossover. Second, reduction and fusion of gametes. And third, independent assortment or random assortment that will cause different type of assortments. So as you are seeing in this video, you can imagine that why our sister is not looking exactly like our mother, and the reason that why we're not looking exactly like our father or like our brother completely 100%, you will find the answer for this question in following slides. So let's talk in details about meiosis. As we defined that meiosis is going to happen inside the gonads of male and female, or it is going to be happen inside the reproductive organs of the male and female organisms. So we divided also meiosis into 2 substeps, that meiosis 1 and meiosis 2 were termed. So let's talk about meiosis 1, about homologous chromosomes separate. In this picture, you're gonna see on the interface chromosomes duplicated. It means this is the cell when it has passed the G1, S and G2 phases in order to enter the M phase. So as you can see, the DNA is in the shape of chromatin. It means it is not condensed, that we should name it chromosome. And the nuclear envelope is also existing, and also the centrioles are at their specific place. Let's talk about that which kind of metabolic reactions is going to happen inside prophase 1, metaphase 1, and anaphase 1 that leads the cell to the meiosis 2. So in prophase 1, as you are seeing the exact features that most of the differences are going to happen from normal cell into prophase 1. The first thing that you see is fragments of the nuclear envelope. It means in prophase 1 the nuclear envelope is going to diminish. Another important thing is this, that chromatin is exchanged into chromosomes. It means the condensed form of the DNA that you can see it with a light microscope. So what is going to happen in prophase 1 is this. The inner membrane disintegrates. As you are seeing, these fragments are fragments of the nuclear membrane. Also, centrioles move to opposite poles. Here you see the centrioles, and in this phase of cell cycle, you can see that centrioles are going to different poles of the cell, and also they are producing the spindles that in further division step it will attach to the centromere or center of chromosomes to split the sister chromatids. Also, homologous chromosomes join synapses, forming tetrads. One of the most obvious differences that is going to happen inside the meiosis that will not occur in mitosis and results to produce genetic variations is the synapse process. In this side, as you are seeing, two chromosomes or two non-sister chromatids are attached to each other. So they are physically exchanging some genes with each other. This is called crossing over. So it means after that sister chromosomes or sister chromatids attached together, they will exchange physically some genes in order to produce genetic variations or variations in genetic. In furthermore steps, you will face that in metaphase, the spindle microtubules attached to canthocores. These are canthocores that are attached into these black dots. These black dots are called as centromeres. That is the attached point of the two sister chromatids inside a chromosome. So also in metaphase, you can see this plate that is called the metaphase plate. It means in metaphase the chromosomes are aligned along the center of the cell. So in furthermore step of cell division, when we face the anaphase, we can see that sister chromatids remain attached, but they are moving toward different poles of the cell. So homologous chromosomes separate from each other. As you see here, these are homologous chromosomes, but in here the sister chromatids remain attached to each other, but the chromosomes will split. So let's talk about the meiosis 2. And in meiosis 2, most of the things are similar to each other, but a few things are going to happen that we will assign those main points. One of those main points is this, that chromosomes were copied inside the prophase 1, but the DNA or genetic material is not going to copied in this phase or prophase 2. But when we phase metaphase 2, the similar things happen. As you see, the chromosomes are aligned at the equator or center of the cell. Spindle fibers are attached with the centromeres. Also, the centrioles are at their exact place. It means the two pole of the cells. And in further step in anaphase 2, you can see that the sister chromatids are splitting from each other, and they are going to different poles of the cell. And the final stage, you will result in haploid daughter cells that they are forming. You're seeing that the nuclear envelope fragments are going to reform again. The centrioles are coming from two pools at their primary place beside the nucleus. So in a result, we can say that meiosis is a kind of cell division which results to produce for haploid cells from a diploid cell. It means they are going to produce unique genetically cells. Thank you so much, dear students. That was the sequence of cell cycle. See you on next videos.